So, find out what happened in the lecture. That sucked. Did you find out what happened? So, just so you know. If we're not specifically in this discussion, will we forfeit our clicker points because they're like not synced?
They won't be synced, but I'm not doing any editing.
Okay.
So, definitely won't be an issue.
With actually your last lecture, I kind of got the gist of what you meant with like being familiar with them. So when you reference something like a cyanobacteria, like we know where it kind of falls in to history. So the other like clades that we talked about, they also have these kind of really, I mean, chloroplasts and mitochondria are really obvious, more obvious because they incorporate into our physiology, or plant physiology. But these other bacteria or eukaryote protists, or proteobacteria is the mitochondria. Right?
Yeah.
Those have different, like significant.
They have tons of different features. The only thing I'm expecting people to know is if I separate from that tour, if we talk about one of those features in some way in more detail, you will be expected to know those features.
Okay.
So if there was something just glazed through in that tour.
Oh no, it's interesting stuff. It's just
no. The point of that tour was not for you to learn all of those details. It was to literally give an introduction. So you don't need to know the material that was in there unless it's referred to again.
Sweet. It is cool stuff.
Sure, but I understand that it's a lot of stuff.
Yeah.
So.
But if I take like a microbiome class, there will be more expected of me in terms of like knowing all of these things for them because it's all about microbiome.
Yeah, that's right. So I want people to like this.
Yeah.
All right. We had some excitement in Section B this morning. I had to evacuate due to a fire alarm. You missed all the excitement. I don't know if anybody heard whether or not there was anything real. It didn't seem like there was anything going on. So maybe there was a test somewhere. All right, so what we're going to do now is mostly talk about Lecture 10, but I'm going to do a little bit of wrapping up the lateral gene transfer discussion that I rushed through at the end of Lecture 9. And then in the next 2 lectures, we're going to be talking about symbioses, primarily microbes that interact with people in positive and negative ways. So just going back to the lateral gene transfer story for a couple of minutes, I went through a couple of model examples about lateral gene transfer. There are hundreds of examples out there of cases where it appears bacteria or archaea or actually occasionally eukaryotes. This is not something that only occurs in bacteria and archaea, but it is more common in them, where one of those groups has undergone some acquisition of DNA from another group. In most of the cases that we know about, there has been very, very strong selective pressure for some particular feature in that environment. The example I started out with was antibiotic resistance. So if you take an antibiotic that kills a particular group of bacteria and you expose those bacteria to the antibiotic, some of the bacteria in that environment might naturally have mutations that allow them to be resistant to that antibiotic. They will survive and then reproduce and spread their traits to their offspring. It is also possible for organisms to acquire the genes that code for various mechanisms of antibiotic resistance from other taxa. And because the selective pressure here is so strong, you are dead, in essence, if you don't have the mechanism to resist the antibiotic. This actually turns out to be a very commonly observed phenomenon with bacteria acquiring the mechanisms to be resistant from other species by taking up their DNA. But we also know many examples where the selective pressure is resistance to desiccation or ability to degrade some compound or a variety of other features. In the E. coli example that I briefly mentioned, where people have looked at different strains of E. coli and have found them to share a lot in common, but also have unique features that appear to have been acquired in one strain of E. coli, not the others, via lateral gene transfer. We don't always know what the selective pressure is behind some of these acquisitions of foreign DNA, although in some cases the foreign DNA allows a particular strain of E. coli to be more virulent or to survive longer on surfaces, say kitchen counters or so on, and that acquisition has been mapped to that particular trait. But in many cases, all we observe is that a particular bacterial strain has some DNA that appears incongruent or different in its evolutionary history than most of the genes in the genome. And so we have a suggestion of lateral gene transfer, but we don't always know what the explanation behind it is. And again, I sort of went through this quickly at the end of class, and I've reorganized the slides a little bit to have them make a little more sense, I think. So in transmission of traits in bacteria and archaea, the big picture is that most, if not all, bacteria and archaea reproduce by binary fission. The cells copy their genome and divide, and they don't have sexual reproduction in the same way that eukaryotes do. And we'll talk more about some of the different aspects of meiosis and gamete production and sexual reproduction in eukaryotes when we talk more about fungi, plants, and animals. And what I'm going to do is go through just a couple of those slides that I went through before. Again, here's the general model for binary fission. In bacteria, the same general process occurs in many archaea. And if you show this in a tree-like structure, this is not an evolutionary tree. This is a model of the cell inheritance, starting with a single cell splitting by binary fission into two descendant cells, each of those splitting into two, each of those splitting into two, and so on. Those can undergo mutation during the course of these splitting events, such that some of the offspring are different than their parents and also different than their siblings. Mutations arise at a reasonably high rate in many organisms, maybe not every generation in every cell, but in a big population you'd see a reasonable number of mutations. And again, if you exposed a bacterial population that had been undergoing a lot of binary fission to an antibiotic, it might be that most of those bacteria would be killed, but a few of them would have randomly gotten mutations that allow them to survive that antibiotic, and those ones would be then transmit their traits to their offspring. You have what I was just trying to show here is differential survival and maybe accelerated growth rate. So in this lineage, the cells are dividing a little more rapidly than in this lineage over here and producing more total offspring. And what lateral gene transfer is very simple in concept. You have different lineages. Let's imagine rather than cell populations, these are now speciation events. So cell division events multiplied by thousands to tens of thousands to hundreds of thousands, if not more, years. And you have different lineages. They accumulate differences by divergence, just like any other species will. And the sort of strange thing about bacteria and archaea, and again, occasionally in eukaryotes, is that DNA can move from one of these lineages to another, even if those lineages are not very closely related. So you may be familiar with sexual reproduction in some eukaryotes where closely related species can occasionally hybridize. Sometimes those hybrid offspring are sterile and therefore don't transmit traits to subsequent generations. Sometimes they're actually viable and do transmit traits to subsequent generations. But in every case, the hybridization events are only between very closely related species or subspecies or organisms that are really close, basically. But in bacteria and archaea, this DNA movement can occur across vast evolutionary distances. You can have bacteria taking up DNA from eukaryotes or from archaea. You can have archaea taking up DNA from bacteria or eukaryotes, and all possible combinations, DNA moving across incredibly large evolutionary distances. And in most cases, when that DNA movement occurs, the DNA that gets brought into a new cell isn't going to work very well. Different organisms have different ways that they carry out some of the cellular processes involved in using DNA. They have different regulatory processes. They even can have different we've already heard about different GC contents of their genome. And the DNA won't work perfectly in every recipient. In fact, in most cases, it's probably not going to work very well. But occasionally, again, especially if there's very strong selective pressure, like for survival in the face of an antibiotic, the DNA that moves can work. It may not work very well at the beginning, but it may produce enough material, say a protein that degrades an antibiotic, such that that strain is going to be a little bit more resistant than its neighbors. And eventually, over evolutionary time, that can get improved upon by mutation and selection until the new recipient strain is highly resistant to that particular antibiotic. So again, the DNA can move across large evolutionary distances. It's not like sexual reproduction because it's usually unidirectional. It's going from a donor to a recipient rather than the merging of two lineages. And this movement of DNA is known by two terms, lateral DNA transfer, lateral gene transfer, or horizontal gene transfer. That is basically describing the opposite or the mirror image of vertical inheritance. So vertical evolution or vertical inheritance is transmission of traits from parents to offspring. And here we have transmission of traits across lineages, so it's horizontal or lateral gene transfer. And then once that happens, this organism here, which had DNA from its parent as well as DNA from this donor, it then transmits both of those DNAs, the donor DNA and the parent DNA, to its offspring. It is now a hybrid. So this lineage here now has two evolutionary histories. And that's why I'm comparing it to the evolutionary history of organelles, where if you have an organelle, a bacteria moving to become an endosymbiont inside another cell and eventually becoming the mitochondria or the chloroplast, that also creates these hybrid organisms. And again, we haven't talked about it a lot. I talked about it in the first lecture or two. We don't show all of the recombination that occurs in sexually reproducing eukaryotic populations. It's one of those features that we gloss over when we draw phylogenetic trees of species. But there's lots of movement of DNA within species via sexual recombination in eukaryotes. And you also, in bacteria and archaea, we won't talk about it that much, you do have, in essence, recombination within species. When the DNA is moving around within a species, we generally call that recombination. When the DNA is moving from one species to another, we call that lateral gene transfer. Any questions about this before we move on into extremophiles? Right. Oh, and again, in chapter 26, there's a little text that you should read on lateral gene transfer, and it shows you a little bit about what the effect is in terms of a phylogenetic tree of different parts of the genome of the occurrence of lateral gene transfer. And it's conceptually basically the same idea as what happens with organelle evolution. Yeah? Is recombination another way of vertical evolution? Recombination is really a form of lateral movement of DNA outside of vertical inheritance. And in sexually reproducing eukaryotes, there's a specialized process called sexual recombination, where we have two, you have parent cells, parent organisms or cells, depending on if it's multicellular or single cellular, producing gametes, those gametes fusing, and those fused gametes can then produce a diploid offspring. So you have basically the equivalent of, it's like lateral gene transfer, but it's now fusing of DNAs from two different individuals, usually within a species. So sexual reproduction doesn't work very well for many reasons between species. You occasionally get hybrids, but usually it's within species events. And the lateral gene transfer events that happen in bacteria and archaea and occasionally other organisms, if that occurs across larger evolutionary distances, we call it lateral gene transfer. If it's movement among different E. coli strains within a species, for example, we're just going to call that recombination. It's not sexual recombination, but it's mixing within a species as opposed to mixing between species. Does that? Okay. So we're not going to go into a lot of detail on this, but if you're interested, I just thought I would show you a couple of examples. So in bacteria, there are many different mechanisms by which they can acquire DNA from other organisms. So one of them is called competence. It's incredibly simple. Bacteria just takes up DNA from its environment. And occasionally that DNA can get integrated into its genome and then transmitted to its offspring. So when cells die, they leak out DNA. That DNA gets eventually degraded, but it can stay around in the environment for a reasonable period of time. And some bacteria can just soak up that DNA from the environment. They actually have highly regulated processes to do that. Many of them may be doing this most of the time just to eat. DNA is an organic compound with all sorts of components that are useful to organisms, but occasionally that DNA can get integrated into the genome and become merged with the cell's lineage. And only certain species of bacteria and archaea are what are called naturally competent, able to do this process. There's a second mechanism which is called conjugation, and that's basically the equivalent. It's a type of mating process that occurs between a donor and a recipient cell. This happens sometimes within species, but it can happen across large evolutionary distances where there's a bridge formed between the cells called a pilus, and then DNA can get sent from one cell into the other cell via this bridge. You even have cases where this can happen between bacteria, for example, and eukaryotes, a form of conjugation transferring DNA into a new host. And then the last major process by which lateral gene transfer is known to happen is something called transduction. And this is basically a case where a virus takes DNA from one host. And when viruses are packaging up their DNA, so a virus will infect a host, it'll take over the cellular machinery of the host, get the host to make lots of copies of its DNA, and also get the host to make lots of capsules, the package that a virus uses to package its DNA. And if everything works perfectly for the virus, the packaging of DNA into the virus will only package DNA from the actual viral genome that was originally sort of inserted into the bacterial cell. But occasionally, the wrong DNA gets packaged into a virus particle. And a virus can actually, the virus doesn't know generally how, some viruses can sort of self-destruct if they have the wrong DNA, but most of them will just take any DNA and then go infect another cell. And so by doing this, a virus can accidentally take DNA from one host and transfer it into another organism, even a distantly related organism. And this is known as transduction. So those processes are found throughout sort of the diversity of bacteria. Many of them are also found in archaea. There are processes like this that do occur in eukaryotes. They're just a little less common, so we usually focus on bacteria and archaea as the hallmark examples of lateral gene transfer. So now we're going to switch over to the second part here, talk about two topics, basically, which are extremophiles and the biology of some of these extremophiles, as well as the methods that we would use to study particular microorganisms. And we're going to focus on using these methods for extremophiles, but these methods would apply to any type of microbe in general. So first, I'm just going to give you a quick reminder about, I mentioned some extremophiles previously. If you remember, we talked about that there's incredible diversity across bacteria and archaea and microbial eukaryotes in terms of their general structures, in terms of many of their functions. And one of the interesting aspects of this diversity is that there are many microorganisms that can survive and even thrive in particularly extreme conditions, including high and low pH, high and low temperature, high salt conditions. And if you made a plot of the ranges of a particular environmental condition at which organisms are known to survive, if you plot, temperature, most of the organisms on the planet that we know a lot about live in sort of a relatively narrow range of temperature. But there are organisms that live at the cold end of this distribution curve and at the hot end of this distribution curve. If they live and thrive at the ends of these distribution curves, they're called extremophiles. It's just the extreme edge of conditions where organisms are known to survive and thrive. And we do the same with pH, the same with temperature, the same with pressure, and so on. And I'm going to just talk about two of these extremophiles here, basically, but there are lots and lots of examples of this. So in terms of studying microbes, one thing that you can do is go out into the environment and try and study the microbes directly in an environment that you go to, say an ocean water sample or soil or human skin. And when you're doing this, in fact, when you're doing just about anything with the original sort of observation about microbial diversity, most people ask basically two main categories of questions. Who are they? That is, what kinds of microbes are found in these environments? And then, what kind of microbe is sometimes informative as to what it's going to be doing, but in general, it's not always informative as to what they're going to be doing. So we also ask, what are those organisms doing in the environment? What functions do they carry? What processes do they possess? And again, we're going to focus on extremophiles, but people ask this about every other type of environment on the planet. So I'm going to go through three general methods in reference to extremophiles that we would use to study microbial diversity. Field observations, going out into the field and studying them. Culturing, that is growing in the lab, and something that we'll talk about at the end, which is called culture-independent studies, or I like to call it CSI microbiology because it's sort of like forensics. So field observations are an incredibly important tool in studies of microbes. We can go out into the field. So backing up, he's not looking for microbes there. So field observations are an important tool in studying any type of organism. We may be most familiar with those in terms of studying plants and animals and other macroorganisms because the field observations are easier. This is Vladimir Nabokov, the author who was also a curator of Lepidoptery at Harvard. He was a big butterfly expert. you can go out and collect, you can go do quadrats, organize sampling in the field. You can use satellite imagery to look at tree patterns, or now people are using Google Street View to look at tree distribution patterns. You can do all sorts of other organized sampling. This poor kid is looking for the bird that's on his head. We can use special tools like binoculars, or we can go to places that we wouldn't normally see things if we have special ways of getting into those environments. Everybody's sort of familiar with these approaches for plants and animals and multicellular fungi and so on. The same general approaches can work for microbes. So you can go out and collect samples. This is someone going and collecting from a Yellowstone hot spring. We can go out into, do transects across large ocean water surfaces. Satellite imagery can be used, especially with wavelengths that particular molecules found in microbes are absorbing and reflecting. Again, we can do organized quadrats. We can use, instead of binoculars, we can use microscopes, and there are many tools where we can go with submarines and other equipment to look in places where we might not normally go. And you can do field experimental studies and field observational studies of microbes in concept in the same way you could do it for multicellular organisms. And the problem with this is generally goes back to this problem that I've talked about many times, which is this work is great and can be very powerful, but it is limited in part by the lack of phenotypic informativeness of microbes as to what they are and what they are doing. So if you go out into a field site and you observe, a large furry organism with huge teeth going around and eating gazelles, you can guess what that organism does in that environment. And we can't do the same thing with many types of observations of microbes. And so what many microbiologists for many years have resorted to is doing some field observations, but then saying, we need to supplement these field observations with more work back in the laboratory. And what people do when you do this is something called culturing. So we can go out into an environment, take a sample from that environment of the microbes, bring them back to the laboratory, and rear the microbes in the laboratory, grow them in a way such that it makes it easier for us to actually do biological studies of those microbes. So culturing is generally known as cultivation, and this is the growth of microorganisms in controlled or defined conditions. So you mix together some vat of, food for a particular microbe, you sterilize it, you put it in certain temperature, and then you add a sample from your environment and you see if you can grow particular microbes in that environment. Now, you can do that with a big batch of soil or a big swab from the skin from any environment. That you can get stuff to grow, just like if you leave out a carton of milk, stuff will grow in it, or if you leave out other food sources. That's not the most useful thing for most microbiological studies. What people really want is what's called a pure culture, which is basically to get a single cell of a microorganism isolated from other cells, and then allow it to reproduce in the laboratory such that it's reproducing asexually, and you're getting it to grow, if it's a bacterium archaea, by binary fission, if it's a eukaryote, by mitosis. It's just making copies of itself in the laboratory. And that making copies gives you material that you can then use to do all sorts of experimental studies of that particular organism in the lab. So basically, the general protocol is you collect a field sample, you provide some specific growth conditions in terms of energy, electrons, carbon. That sound familiar? Certain types of parts of the nutrition of the organism. You might have to modify other conditions like the temperature and the oxygen and the salt, and you go through many generations where you're trying to dilute out the organisms basically until you have a single cell that is able to grow in the laboratory. And there are many different approaches to doing this. You can do this on a plate. I assume most people are familiar with things like this. You can do it in test tubes. You can do it in a variety of other situations. And eventually you get to the point where you have a lot of material and you can reproduce it regularly and you can grow that particular isolate and do experiments on it. And culturing has revolutionized microbiology. If you're interested in a particular function or a particular microbe or a particular environment, it's really important to figure out a way to grow those microbes in the laboratory and do experiments with them. And there are many different reasons for doing this. I'm just going to go into some of the reasons. So for example, if you grew microbes in the laboratory and you wanted to figure out who was out there, that is, what microbes were present in the original environment. You go to that environment, the appearance isn't that useful, but you can bring them back to the laboratory, grow them in the laboratory, and then even though the appearance might not be useful, you can apply the same methods that we've talked about in the course where you can isolate ribosomal RNA genes from that microbe and say what kind of organism it is. So we can take that same approach that Carl Woese did and take the microbes that you were growing in the lab, and that's basically what he did for many of those organisms, isolate the ribosomes, isolate the ribosomal RNA, read the sequence, build a data matrix, build an evolutionary tree, and wherever your new organism sits on this evolutionary tree will tell you what kind of microbe it is. If it's a cyanobacterium, it should group in the evolutionary tree with ribosomal RNA genes from other cyanobacteria. If it's a spirochete, it should group with ribosomal RNA genes from other spirochetes, and so on. That's how you tell who was there by rearing something in the laboratory. And if you want to figure out what they are doing, you're basically asked what are they capable of doing by studying them in the laboratory. And here I'm going to give you a couple of examples of this. So suppose you were interested in the microbes that were in these very salty environments. This is salt evaporators that I've mentioned before, where the salt concentration is going up and up as the water evaporates and different microbes are growing in these environments. And suppose you got a microbe out of this environment, you were able to grow it in the laboratory, and you wanted to know something more about its biology. Here's a protocol by which you could figure out a little bit more about its biology. So you culture that particular organism, you take a single clone, a single cell basically of that organism, and you grow it up, and then I'm going to show you this visually too, this is the text version. You put a little sample of that microbe in different flasks. So hopefully that microbe, everything you're putting in the different flasks is genetically identical to each other. And the main difference you're going to get is how this organism grows in these different environmental conditions. So you could vary the salt concentration or vary the temperature. And then what you do is you watch as the microbe does or does not grow in those conditions. You can monitor whether or not it's making copies of itself by looking at basically the density of the culture. The more cells that are in there, the harder it is to see through it in many cases. And you can just monitor the change in growth of this over time. So here's a visual example of this. You go to the environment, you isolate something, you grow a little, what's called a starter culture. You have some flasks that maybe have some differences between them. You add a little portion of your microbe to each of those flasks. Let's say one of them has low salt, two of them have medium salt, one of them has high salt. Those are our 4 different conditions. You can do it with different molarities of salt, for example, and everything else being equal. And now we monitor growth over time. Maybe after a little bit, you can see that it's growing a lot in this sample and only a little bit in that sample. You let it grow a little bit longer, and you monitor this over a long period of time, and then you plot at each time point for each concentration of salt what the density of your culture was, how many cells basically were in that culture over time. And you can then convert this plot into a rate of growth of the organisms. How fast are they making copies of themselves? And we can ask, under what conditions does this organism make copies of itself the fastest? So now we're plotting the growth rate versus those salt concentrations. So in a simple experiment, we figured out that this particular strain of organism under those conditions grows the fastest, prefers to grow under 3 molar salt concentration. And it does not grow very well under 4 molar or 1 molar, and it grows a little bit, but not quite as well under 2 molar. So this is how we figure out when we go back to the lab what type of environment organisms prefer to grow on. This improves our ability to actually culture them, and then we can take this and do lots more experiments learning about the biology of these organisms. When people have done this with various salt-preferring or salt-not-preferring organisms, if you plot sodium ion concentration versus one of these growth rate curves, We see, for example, that many organisms like E. coli hate salt. So basically, as soon as you start to add decent amounts of salt to the solution that it's growing in, the growth rate of E. coli plummets. E. coli is what we would call a non-halophile. Halophile means prefers to grow in the presence of salt. E. coli does not prefer to grow in the presence of salt. There are some organisms that are generally classified as halotolerant. They prefer to grow in low salt concentrations, but they can grow in higher salt concentrations. Staphylococcus aureus, that's the species that MRSA is a member of, methicillin-resistant Staph aureus. It likes to grow in low salt concentrations. That's where it grows the fastest. But it's hard to kill it just by adding salt. It can keep growing, basically. And then as we get to higher and higher salt concentrations that organisms prefer to grow at, we start to call those organisms halophiles. That is, they actually prefer to grow in higher salt concentrations. And what we see is something really interesting, which is that the most extreme halophiles, the ones that only prefer to grow in incredibly high salt concentrations, most of those that have been isolated are archaea. They're called halophilic archaea. When people have done experiments on organisms that grow in high salt concentrations, there's sort of some general expectation that you can have. If you put an organism that normally is used to low salt into an environment with high salt, there should be basically osmotic pressure on the cell. The water should want to get out of the cell, and the cell itself will shrivel up and shrink if you put a normal cell in high salt environments. So what organisms that tolerate or thrive in high salt environments do basically is compensate for this in a variety of ways, usually by filling up the inside of the cell with some other compound that basically prevents the desiccation of the cell. There are many halophiles that are in the bacteria and eukaryotes. Most of them don't go quite as extreme as these archaea, but they have all sorts of adaptations that allow them to survive in these higher salt conditions, usually by filling up the inside of the cell with things that are called like proteins and carbohydrates. What's very unusual about the archaea that live in high salt concentrations is they fill up the inside of their cell with salts. So what filling up the inside of the cell with these compounds does is it basically helps prevent them from losing water. And filling up the inside of the cell with salts is very strange because it means that every other component of the cell has to work in high salt concentrations. So this filling up with salts is only done in the halophilic archaea. If we go to the evolutionary tree of archaea, this is a higher resolution one than used in the book. We find that the halophilic archaea are a single monophyletic group. So this adaptation of filling up the inside of the cell with salt happened once in evolutionary time in the common ancestor leading up to the branch of the haloarchaea. And it turns out that many industries are very interested in this because there are many industrial processes or industries that would like to sell certain processes that have to function in high salt concentrations. So laundry detergent is a classic example of this, but there are many other processes that work in very high salt. And what a lot of industries have been doing in the last 30 or 40 years is isolating enzymes from different organisms and adding them to their industrial process in order to make it work better. If you wanted to make degradation of proteins and carbohydrates work better, say for a laundry detergent, and the laundry detergent is filled with lots of salt, if you got an enzyme from E. coli, it wouldn't work in those conditions. If you got an enzyme from just about any bacteria or eukaryote, it wouldn't work in those conditions. But enzymes from halophilic archaea work in those conditions. So you can purify the enzymes from these organisms. Those enzymes work regardless of being in the presence of incredibly high salt concentrations. So this is a source, in essence, of industrial products by going to organisms that have figured out not only how to survive in high salt, but how to do it by filling up the inside of their cells with other salts. So that general approach, where I didn't go through it in fine-scale detail, where you culture an organism, You then grow that organism in the laboratory. You can compare what conditions the organism likes to grow at in the laboratory, find the best conditions for that organism, and eventually what you can do is study the adaptations that organism has to those particular conditions. And for extremophiles, there's been an enormous amount of work trying to figure out how organisms are able to survive and thrive in these particular unusual conditions, both to generally understand the biology of organisms, but also, again, because there are many applied uses of the products from these organisms. So another example of this, I'm just going to sort of quickly go through this, is organisms that grow in high temperatures. This is an overhead view of one of the hot springs in Yellowstone National Park. And in the middle of the hot spring, it's very, very hot. As you get to the edge of the hot spring, it cools off a little bit. And there are organisms that grow at temperatures in Yellowstone up to 90 or so, 95 degrees. They basically grow until the water starts to boil. And at Yellowstone's at higher altitude, so the water boils lower than 100 degrees Celsius. But again, if you do the same general approach as with the halophiles, you go to a hot environment, you culture things, you grow them in the laboratory, you do this experiment where you compare under different growth conditions. In this case, we would compare different temperatures rather than different salt concentrations, monitor growth over time, plot the growth over time, calculate a growth rate. If we do this for different organisms, we now have the spectrum of temperatures at which organisms prefer to grow. And if you do this, you find that some organisms prefer to grow at temperatures that are around human body temperature. Those are many of the organisms associated with mammals prefer to grow at temperatures like that. Some organisms grow at very low temperatures, and some organisms grow at incredibly high temperatures. The ones that grow at high temperatures are called thermophiles. If they grow above about 85 degrees Celsius, that is, they prefer to grow, they're called hyperthermophiles. And amazingly, there are some organisms that actually grow above the boiling temperature of water. Boiling normally will kill organisms. Basically, all organisms don't like to be boiled. But in the bottom of the ocean, the pressure of the water makes the boiling temperature of water higher than 100 degrees Celsius. And there are organisms in the bottom of the ocean at these hydrothermal vent sites that actually survive and thrive and prefer to grow at temperatures above 100 degrees Celsius. If you look at the organisms that are hyperthermophiles, and in particular as you go to higher and higher temperatures within the hyperthermophiles, those organisms tend to be archaea. So the archaea are the ones that grow at the absolute highest temperatures. Bacteria can be hyperthermophiles, but usually only up to about 90 degrees Celsius, not 105, 110. You don't need to look at this whole detail. The main point of this is to say that if you look at different organisms, there are thermophiles that are found in every major branch in the tree of life. But eukaryotes generally can't get up to even hyperthermophile range, and archaea tend to grow, some examples of archaea grow at even higher temperatures than bacteria are able to grow. So if you look at the highest temperatures where organisms grow, those places are dominated by archaea. Bacteria can fill in environments that are really thermophilic, and eukaryotes that are hyperthermophilic. And there are eukaryotes that are thermophiles, but they can't seem to be able to tolerate incredibly high temperatures. So the extreme high temperatures are dominated by bacteria and archaea, and as you get to higher and higher temperatures, you see archaea dominating those environments. Unlike with halophiles, if we plot the distribution pattern of thermophiles and even hyperthermophiles across the tree of life, we see that, shown in red, there are lots of different lineages of bacteria and archaea that are thermophiles or hyperthermophiles. The only plausible explanation for this, and this has actually been shown through many studies, is convergent evolution. In different lineages of bacteria and archaea, they have separately evolved the ability to grow at very high temperatures. Just like with high salt, you can look at the stresses that might occur with high temperature, and by doing experiments in the laboratory, figure out how organisms have been able to deal with those stresses. So growing at high temperature in general should make proteins, RNA, and DNA less stable. The folding of proteins, RNA, and DNA depends upon hydrogen bonds in many cases, occasionally covalent bonds. Those get disturbed as the temperature gets higher and higher. So one adaptation of many thermophiles is different strategies to make their proteins and RNA and DNA fold more tightly even when the temperature is high. Reactions will speed up, so a default enzymatic reaction will occur at a much higher rate as the temperature increases. So actually, one adaptation of thermophiles is to, in essence, weaken their enzymes in many cases so that their reactions don't occur at an incredibly high rate. The membranes of cells should basically be liquefied as you get at higher and higher temperatures. It's basically like heating up butter or some other waxy compound. And many of the thermophiles have special adaptations that basically decrease the fluidity of their membranes. One effect of all of these adaptations is if you put these organisms in cold temperatures, they don't do very well. Their membranes start to gum up. Their reactions now occur at way too low a rate compared to what they're sort of designed to do. And their proteins are basically overly stable, hard to change the structure of their proteins. Are there any questions about thermophiles or halophiles before I go on? All right, so the general idea here is that we can use growth in the laboratory to characterize these organisms. We can identify the adaptations seen in these organisms by comparing, in particular, organisms that have the ability to grow at high temperatures to organisms that don't have the ability to grow at high temperatures, or the organisms that can grow at high salt to organisms that can't grow at high salt. The way we do these comparisons varies. So for the halophilic archaea, they're a monophyletic group. We can really just compare them to one outgroup that doesn't have this adaptation. For the thermophiles, because this ability to do this has shown up so many different times in evolutionary history, you have to be careful because two different thermophiles may have slightly different ways that they have adapted to growing at high temperature. I'm not going to go into all these details, but there's some examples throughout the book of all sorts of other extremophilic environments and some practical uses that come from those extremophiles, whether for industry or food production or producing various compounds for other uses. If you are interested, there's a company in Davis that specializes in doing this exact type of thing called Novozymes. They specialize in going out into particular environments, including extremophile environments, although not only extremophile environments, and isolating enzymes from different organisms to apply them to be used in various industrial, agricultural, and food production processes. And they use a lot of knowledge about the biology of these organisms and their evolution, in essence, to find and identify and characterize these processes. And the last thing I want to leave you with, we'll talk about this more in the next two lectures, is this issue of CSI microbiology, and I'm just going to give you a couple of minutes on this. So if we go to that Yellowstone hot spring, or if we go to a solar salter and evaporating environment, if we go to human skin, or if we go to any other environment on the planet, and we take a sample from that environment, and we split that sample in two, and for half of the sample we try to grow anything we can out of that sample, any of the microbes, and for the other half of the sample we look in a microscope, and we count how many total microbes could we grow by growing them via culturing? And how many microbes could we see by looking at them in the microscope? In every environment that's been looked at, we see huge numbers more, sometimes many orders of magnitude more microbes in the microscope than we are able to grow in the laboratory. This is generally known as the Great Plate Count Anomaly, because to count these microbes that you're growing, you count them on a bacterial plate, a plate of food, basically. And this has vexed people for many, many years. There's been a big debate about what explains this, but it makes it hard to characterize a particular environment. If we can't grow the microbes in the laboratory, we can't say who they are, and we can't say what they're doing. So this has limited our ability to characterize the diversity of life on the planet because, again, environmental field studies are useful but limited in scope. Microscopy, we can't identify the organisms very well from microscopy, and we can't culture most of the organisms from a particular place. The solution that has been developed for doing this has been to go directly to the environment, isolate DNA out of the environment without ever trying to grow anything, and to apply the same methods that we talked about for characterizing microbes in the lab, but now characterizing their DNA in the environment. So you go out to an environmental sample, you isolate the DNA from that environmental sample, say from these giant tube worms in the bottom of the ocean. They were thought to have bacteria that live inside of them. We'll talk more about that later in the class. In microscopy, you can see lots of cells that look like bacteria living inside these giant tube worms. You apply a special method that we will talk about a little more later called the polymerase chain reaction, which is really good for making copies of ribosomal RNA genes in the laboratory from organisms that you've never been able to grow. So you take the samples from the environment, you isolate DNA from those samples, you run this PCR reaction, which makes copies of the genes for the ribosomal RNAs. You read the sequence of those genes just like Carl Woese was reading the actual ribosomal RNA itself. You make a data matrix, you build an evolutionary tree, and now you can place on your evolutionary tree organisms even if you have never grown them in the laboratory. So people are using this general approach, this culture-independent DNA analysis, or again, what I like to call CSI microbiology, to characterize the diversity of microbes on the planet. And what we will talk about in the next 2 lectures is how people use approaches like this to characterize the microbes that live in and on us, which, are of great interest to us. And I will stop there. Yeah.
When you're, do you assign it totally subjectively if you want it transversions to count 2 times or 3 times more? You kind of take into account the situation.
Yeah, you may not have heard this.
So that's a good question.
Yeah, it was a very good question. Some people have asked this in at least one of the sections. So what you actually do is you take data from organisms where you think you know something about the history. Like that phage experiment that I talked about in the book and that I talked about in lecture, or some other experiment like humans, chimps, and gorillas where you think you know the relationships from the fossil record. And then you sequence DNA from those organisms and use ads. Instead of trying to figure out what the rates of transitions and transversions are, you lock the tree, and now you can calculate the rate. You can see how many of the changes on one branch were G to A changes, how many of the changes on another branch were G to C changes. And you can actually measure the ratio. That then tells you the next time you want to do this, what the... Yeah.
Okay. All right, thank you.
Yeah.
With the CSM microbiology stuff,
Yeah.
if you have like a thermophile and you take them to like take out the DNA really fast, does the taking it away from the heat source, does that mess things up?
Not for the DNA. Once the DNA is out of the cell.
It's fine?
Yeah.
Okay. I just wasn't sure if there's some sort of backwards protein malfunction.
We get rid of all of that. We just strip all of that away and look only at the DNA.
Okay.
But it's a very good question because it affects your ability to get the DNA out, but once you get the DNA out,
You're good.
yeah.
Okay.
So you survived the fire.
We're behind.
I sent you a message and I realized you were in a lecture. It's awesome. I have a question.